Hello guys, very warm welcome to all of you. Welcome to our modern biology. I'm Sasmita. Today we will see plasma membrane part 4. I have already covered plasma membrane part 1, 2 and 3. If you haven't checked that video, then go and check that out. Otherwise you will not able to understand this one. So without further delay, let's get into the video. Before that, don't forget to like, share and subscribe to my channel, and don't forget to press the bell icon for further notification. So very first we will see phospholipid. It is divided into following types. We have already know the type of phospholipid in our previous video, but today we will see the types in more detail. I have already told you the types. It is divided according to the alcohol ring in the head group. Then the next one is phosphatidyl ethanoamine. We will see this in more detail. Phosphatidyl ethanoamine PE is also known as cephalin. Now, if you will ask me why it is called as cephalin, because phosphatidyl ethanoamine first isolated from the brain. Because of that, we call it as cephalin. Then the next point is that it is class of phospholipid found in the inner cytoplasmic leaflet of the membrane. Like it is mainly found in the inner cytoplasmic leaflet. Then the next point is it is composing of 25% of total phospholipid. Then if we will talk about the charge of phosphatidyl ethanoamine, then we will see net positive charge aminolipid. Then why we will call it as aminolipid? Because it has amine group which is added in lipid, so we call it as aminolipid. Then we will see its structure. If we will see the phosphatidyl ethanoamine structure, you can see you can find a structure like this. There you can see there is three carbon number 1, carbon number 2 and carbon number 3. Carbon number 1 bind with the fatty acid and carbon number 2 bind with the again fatty acid, and carbon number 3 bind with the phosphate group which is in turn bind with the ethanoamine. Carbon number 1 which is bind with the fatty acid, it is 15 to 18 carbon long which is saturated. And carbon number 2 which is bind with the again fatty acid, it is 18 to 20 carbon long and it is unsaturated. Then if the phosphatidyl ethanoamine present in carbon number 3, we call it as alpha cephalin, and if it is present in the carbon number 2, we call it as beta cephalin. The next we will see phosphatidyl serine. There is no separate name has given and it is found in the brain nervous tissue, that means neuron. And the most important feature of the phosphatidyl serine is it is also present in the blood. Like phosphatidyl ethanoamine and phosphatidyl choline is also present in blood, but very less concentration. But this the phosphatidyl serine is present very significant amount in the blood. Then the next point is phosphatidyl serine is the most the major acidic phospholipid class that accounts 13 to 15 of phospholipid in human cortex. Then the next one is found inner leaflet of lipid bilayer but flips out if apopriosis occur. Like it is found in the inner leaflet, like as I had already said, phosphatidyl ethanoamine also present in the inner leaflet. Like when a cell apopriosis occur, the phosphatidyl serine flips outside, like outside of the membrane. When it goes to the outside, like the outer leaflet, it gives signals to the cell for apopriosis, like it acts as a signaler. If we will talk about the charge, then it is negative charge and it is aminolipid. Like because it has a amine group which is added in lipid, so we call it as aminolipid. So the next point is it plays a key role in cell cycle signaling, especially in relation to apoptosis. I had already said this point before. Then if we will see the structure of phosphatidyl serine, then we will find a structure like this. I had already this structure is quite similar to the phosphatidyl ethanoamine, but we will in this structure we will find a serine backbone with three carbon, like carbon number 1, carbon number 2 and carbon number 3. Carbon number 1 is attached with the fatty acid, and carbon number 2 attached with the again fatty acid, and carbon number 3 attached with the phosphate group, which is in turn with the serine. Remember, this serine is your amino acid, and this structure is quite similar with the phosphatidyl choline. And if we will see the carbon number 1 which is attached with the fatty acid, it is 16 to 18 carbon long, and it is saturated. If we will see the carbon number 2, it is attached with the fatty acid which is 18 to 20 carbon long, and it is unsaturated. And the bond between over here is ester bond. Now we will see phosphatidyl choline. It is also known as lecithin. It is most abundant phospholipid in animal tissue. Like lecithin is present in everywhere, found in the outer leaflet of lipid bilayer. Then the next point is carinate positive charge and absent in bacteria, as well as incorporate in the head group choline to form phosphatidyl choline. If you will see the structure of phosphatidyl choline, then you will find glycerol molecule with 3 carbon, containing glycerol molecule which is contain 3 carbon, carbon number 1, 2 and 3. Carbon number 1 first bind with the like fatty acid which is 16 to 18 carbon long, and carbon number 2 bind with the fatty acid again, and it is 18 to 20 carbon long, and carbon number 3 which is bind with the fatty acid which is in turn bind with the choline group. And the carbon number 1 which is bind with the fatty acid, it is saturated, and carbon number 2 which is again bind with the fatty acid, it is unsaturated fatty acid. And carbon number 1 and carbon number 2 bind with the fatty acid by ester bond. And this reason is a polar group, so that it is not always present in the third carbon, it is also present in the second carbon. You can see over this structure. So in this structure we call it as lecithin. If it is bind with the carbon number 3, we call it as alpha, and if it is bind with the carbon number 2, it is called as beta lecithin. It is alpha lecithin and it is beta lecithin. Now we will see asymmetry of lipid bilayer. The phospholipids in the plasma membrane is asymmetrically distributed in the plasma membrane. In this, we will know which lipid is present in which location of the plasma membrane. We call it as asymmetry because it does not have any fixed uniformity, because division occur asymmetrically. So we call it as asymmetry. And the amino containing phospholipid are enriched in the cytoplasmic surface of the plasma membrane, while choline containing and sphingosine in the outer surface of the plasma membrane. And the lipid asymmetry is essential for the maintenance of transbilayer diffusion, like lipid protein interaction and lipid mediated transportation. Then if you will see this structure in more detail, then you will find here 100% of phospholipid, and you can see over here is the total phospholipid. Then if you will see PC means phosphatidyl choline, you will find the it is present in more in the outer side than the inner side. It is present less in the inner side. And PE phosphatidyl ethanoamine, it is present in more in the inner side than the outer side. It is present less in the outer side. And then if you will see PI means phosphatidyl inositol, if you will see these, then you will find equal amount which is present in the outside and inside, both are the same amount. PI means phosphatidyl inositol. You have already known that it is very important signaling molecule, like 4,5 phosphatidyl inositol on IP3 pathway is signaling molecule. So signaling, so it is very important for signaling, like for signal. Then we will see PS means phosphatidyl serine. It is solely present in the inner side of the plasma membrane. You can see over this structure. And as I had already said you that it act as a apopoietic like signaler. Like when apoprocess occur, it flips out from the flips out to the outer plasma membrane and act as a apoprotic signal, like apoptotic signaler. If you will see the SM, SM means sphingolipid. We call we wrote I wrote it here SM. It is mostly present in the outer side of the plasma membrane. Like we see over here, the excessive amount present in the outside, we consider it present in the outside. And the most excessive amount present in the, if the most excessive amount present in the inside, then we call, we know it as present in the inside of the plasma membrane. In the PS and PE present in the higher amount, and sphingosine amount in the outer surface is equal to the PE in the inner surface. So guys, it was our fourth part of plasma membrane session, and I hope you all had a good session. Thank you so much for watching our video, and thank you so much for your love and support towards me. Keep loving and keep watching.